Fantastic. All right, so good afternoon to all. I hope that you had a great summer, first and foremost. And I would also like to welcome all the new trainees who may join us for the very first time for the QCRI seminar series. Today, we're fortunate enough to have Dr. Simon Turcotte as a speaker. Dr. Turcotte is an associate professor at the Department of Surgery of the University of Montreal. He holds the Roger Desgroseilliers Chair in Hepatobiliary and Pancreatic Oncological Surgery. He completed fellowship training at the surgery branch of the National Cancer Institute in Bethesda, Maryland, with Dr. Steve Rosenberg. And he also completed surgical subspecialty training at the Memorial Sloan Kettering Cancer Center in New York. Most notably, after showing the spontaneous T-cell recognition of GI cancers in vitro, he launched a first trial using tumor-infiltrating lymphocytes immunotherapy for GI cancer patients as a postdoctoral fellow at the NCI with Steve Rosenberg. He is also one of the very few surgeon scientists supported by the Quebec-based FRCSC, and administers a large biobanking effort to run a translational research program focusing on how to improve T-cell immunotherapy for common solid cancers. He is currently supported by BioCanRx and is the PI of the very first T-cell immunotherapy trial to be conducted for solid cancers in the province of Quebec at the CHUM. On perhaps a more personal level, Simon is also one of the very first individuals with whom I had a chance to discuss a potential research-oriented career. So, Simon, I'll always be grateful for the time that you had given me back then, talking about opportunities and immunobiology. Simon actually became one of my PhD thesis committee members a few years after. So again, it's a real pleasure to reconnect and have you this afternoon. So we're all looking forward to your talk.
Great, thank you for this very kind introduction, Pierre-Olivier. I'm really pleased to be a speaker to the Queen's Cancer Research Institute seminar series today. I'm going to share my screen here, just confirm that you can see it. Can you see the screen, everyone? You can hear me as well?
Yep.
Yes.
All right, perfect. So I've titled my talk Adoptive Immunotherapy with Tumor-Infiltrating Lymphocytes, Developing New Cell Products and Initiating a Clinical Program for Solid Cancer Patients. My research is funded by, and partly by Iovance Biotherapeutics, Center and Zone and Bioethics, my disclosures. New treatments are definitely needed for patients with solid cancers. You know, it's the first cause of mortality in Canada, cancer, and 90% of patients die because they have a solid cancer that is metastatic and refractory to standard treatment. You've all heard that cancer immunotherapy comes as a revolution in the field of oncology, and this is largely based on the ability of T cells to recognize, just as this T cell you see in a dish, to recognize a cancer cell, lyse this cancer cell, destroy it in the body. This T cell is a very powerful tool. T cells are able to recognize heterogeneous solid cancers because these cancers are mutated. As you know, there is stochastic mutation in the cancer cell that eventually encodes for proteins that are degraded, and mutated peptides can be presented by MHC molecules at the surface of cancer cells for then be recognized by T cells that are patrolling the body and infiltrating tumors. These tumor-infiltrating lymphocytes, which we refer to as TILs, are polyclonal. They're reactive to potentially multiple neoantigens. They are functionally heterogeneous. They are autologous. They underwent thymic selection, and this is very important because you shouldn't expect that a tumor-infiltrating lymphocyte recognize self-antigens with high affinity. And so for therapeutic use, this offers a lot of advantages. And they can recognize neoantigens, which are cancer exclusive, not expressed in normal tissue, and they're patient specific. So allowing for a very highly personalized and cancer specific therapy. Neoantigen reactive T cells are enriched in tumors. They're nearly undetectable in the peripheral blood. This is established by multiple publications over the year. I'm just showing a snapshot here from the published literature and an example from a case we've studied extensively. So these are two melanoma patients, and Cyril Cohen, who, when he was at the NCI, he's now a scientist in Israel, had used tetramers to identify TILs, tumor-infiltrating T cells, that can recognize mutated neoantigens predicted from the sequenced tumor. You can see in this tumor cell suspension, in this melanoma tumor cell suspension, you have 1.1% of TILs that recognize mutated AHN-AK gene, 5.6% recognizing this gene, 1.9% recognizing this gene. If you use the same tetramers and you try to measure recognition of these neoantigen peptides from circulating blood, you see that the percentage is nearly zero. It's another example here from another melanoma tumors. Spontaneous immune recognition of solid cancer is not restricted to melanoma, even though melanoma has higher mutational burden because of UV carcinogenesis. You can find neoantigen reactive T cells in most solid cancers, even in low mutation burden, just like this liver metastases from a patient with metastatic colon cancer, in which we found neoantigen TILs that recognize 4 different mutated genes. Those T cells bear different TCRs recognizing the same mutated gene with different affinity. And you can see that the highest percentage of neoantigen reactive cells in this liver met is of about 5% or so. You have other TCRs that are present at low frequency that recognize these neoantigens. But prior to surgery, those neoantigen reactive T cells are not detected in the blood. Several years after surgery, we may be able to detect at low frequency some of them. But truly, those neoantigen reactive TILs are present, are enriched in solid tumors and are really difficult to detect in the peripheral blood. We went back in time for this patient and used sequencing to test whether we could detect neoantigen reactive T cells in the primary tumor, which was the case. In metastatic nodes, initially there were no reactive T cells. When this patient relapsed, there still was some tumor reactive T cells detectable in its tumor, but not in, well, and in some also in metastatic nodes. In this case, we didn't have the blood to show that those T cells were barely detectable in the peripheral blood, but I assume it would be the case. So tumor is the best place to find the highest possible percentage of tumor reactive T cells. How can we use those tumor reactive T cells for therapy? This effort started really at the NCI, initiated by Steven Rosenberg in the 80s after IL-2 was cloned and available as a reagent to work with T cells in vitro. And he first showed in a mouse model with a surgeon from actually Quebec, René Lafrenière, who was there doing his postdoc at the time, that TILs could be expended from implanted MC38 syngeneic colon cancer tumors, and that when those TILs were administered after a lymphodepletion and in combination with IL-2, 100% of these mice could be cured by the therapy. And this really became the classic scheme of TIL therapy, where those three components, the lymphodepletion, the T cells, and the IL-2, are considered key to get the highest efficacy for TIL therapy. Another thing that I wanna point out from this early study is that prior to testing tumor-infiltrating T cells, Dr. Rosenberg and his team was investigating LAC cells, T cells derived from the peripheral blood of patients and of animals that were activated with high dose of IL-2 in vitro and re-injected to patients and animals in this case. And you see that even if you take peripheral blood and you inject it after lymphodepletion in combination with IL-2, there may be some tumor control, but translated here in median overall survival that is slightly prolonged, but certainly no cure, as was the case with TIL. So based from these early studies, the TIL therapy was tested in human from the 90s and on, and the classic scheme became the following. The patient has to undergo tumor resection of a metastasis with minimally invasive surgery so that the patient recovers fast from the surgery. The tumor is transformed into small pieces or cell suspension, put in culture with high dose of IL-2, human serum, eventually rapid expansion protocol with feeder cells and OKT3, as to produce a large amount, billions of T cells ex vivo outside the body. Once there is enough T cells, the patient is readmitted to the hospital, receives non-myeloablative chemotherapy with cyclophosphamide and fludarabine, a 5 or 7 days course of chemotherapy. Once his white blood cell count is down to 0, the patient receives the TILs, and this is followed by adjuvant interleukin-2 to support the adaptively transferred TILs. What's the efficacy of this treatment? The largest series, the largest number of patients have been treated at the NCI, and the effort started with metastatic melanoma. So it's really with these patients that we have the most robust data. And in this article published in 2021, we see the update results from 2026 melanoma patients who received TIL. Half of them showed an objective response rate, complete response. Complete response, no more visible metastatic nodules were seen in 22% of these patients. And from these 49 patients, only a few patients, as you can see here, relapse after complete response. So those complete response were highly durable, as you can see with the long actuarial follow-up here. And the median survival in this cohort of patients was of 22.2 months. that's the experience and data in metastatic melanoma at the NCI. It's now also supported by meta-analysis from data published by other centers. Now there is a large biotech called Iovance Biotherapeutics that is running multi-center trial with central manufacturing of TILs and start to publish results in metastatic melanoma and other cancer types. There is a strong positive signal that in end-stage metastatic lung cancer, TIL therapy can be effective in about 20% of patients. There is also a strong efficacy signal in patients with cervical cancer. Some case reports for other solid cancer like breast cancer, and the one we had published in a bile cancer patient who we initially treated about now 10 years ago and who's still alive after TIL therapy. This patient, I want to point out, became an advocate, a cancer advocate, and an advocate for TIL. Her name is Melinda Bacchini. She publicly speak about her what she underwent and TIL therapy. She had a low mutation burden tumor. We only found about 35 mutation in her tumor, but there were those very neoantigen reactive CD4 T cells that we could expand ex vivo and reinfuse to this patient who had large metastatic disease burden in the lung, and again, who's still alive 10 years after her treatment. TIL therapy is developed in many academic centers, a growing number around the world. Toronto has been the first in Canada at the Princess Margaret with Pam Owashi and Marcus Butler to provide access to melanoma patients to TIL therapy. And there's a growing number of companies that are now running multi-center trials with various TIL products and various indications. The nearest indications for FDA approval now is considered melanoma, cervical cancer, and lung cancer due to trials led by Iovance Biotherapeutics. So let's just step back a bit. You've heard of cancer immunotherapy. You've mainly probably heard of immune checkpoint blockade. Immune checkpoint blockade can only work if there is a spontaneous, endogenous, sufficient anti-tumor immunity against solid cancer, which unfortunately is not the case for the majority of patients. Immune checkpoint blockade can be highly effective in melanoma, in highly mutated tumors, but this still represents a two-liter subset of patients. The goal of adoptive T-cell therapy is really to amplify ex vivo this anti-tumor T-cell response. The entire scheme offers the possibility to prepare patients for TIL by giving immune preconditioning. This lowers the regulatory mechanisms that impairs T-cell immune recognition. And once the cells are given to patient, there is still an opportunity to boost this T-cell response by giving immune checkpoints, for instance, and IL-2 as commonly done in these protocols. And these combinations are under study in many trials, the combination of immune checkpoint pre and post TIL infusion. Adoptive T-cell is one treatment, it's hospital-based, so obviously it's resource intensive, but we do bone marrow transplant in the public health system in Canada. We do solid organ transplant. We do many things that are more complicated than adoptive T-cell therapy to a large number of patients. So I wouldn't see this as an obstacle for development of this therapy. And again, the efficacy signal is positive in many indications, and trials are ongoing. All right, so I told you what TILs are, what's the general scheme of TIL therapy. Now, what we're doing in the lab is to try to try to increase the efficacy of TIL and T-cell therapy for solid cancer patients. And the main challenges that we face is that even though I told you that the tumor is enriched in neoantigen and tumor-reactive T cells, it still remains a low percentage of infiltrating T cells that recognize tumor antigens. You saw in the previous graph 5% of T cells that recognize neoantigens. So we need to find ways to enrich and select those tumor-reactive T cells to make better cell product, more reactive T cell product. The other problem is that if you use the standard bulk expansion of T cells, the tumor-reactive T cells often are overgrown by bystander T cells. Those bystander T cells may often have a better proliferative capacity than a T cell that has repeatedly seen its antigen, its tumor antigen in vivo and submitted to immunosuppressive mechanisms in the tumor. And so bulk expansion of T cell is not ideal in this treatment scheme. So facing this challenge, we are trying to develop ways to select reactive T cells. And the biology we are investigating is based on the fact that a T cell that repeatedly see its antigen do express high level of PD-1. As simple as this. PD-1, you know this protein because anti-PD-1 therapy is the most common nowadays immune checkpoint blockade therapy. But PD-1 is also a tag for tumor-reactive T cells in a tumor. We've shown in 2014 in preclinical work with Elena Gross that we could select CD8 T cells that express a high level of PD-1, expand those cells, and showed in vitro that this was a great way to enrich in neoantigen and autologous tumor-reactive T cells. And another group has shown, in mouse model, here reference number 4, that this strategy was more efficacious than using bulk TIL to treat mice with implanted tumor. And so 5 years ago or so, we decided we had the chance to be supported by our foundation at the Cancer Access here, Institut du Cancer de Montréal, that allowed us to acquire a clinical grade cell sorter to try to translate this TIL selection scheme into a clinical application. And the idea is that we start with a resected tumor, transformed into a cell suspension, stayed with antibodies, fluorophore conjugated antibodies, just like for FACS analysis, enabling us to identify the CD3 positive T cells within the tumor cell suspension that express high level of PD-1. We place those labeled cells in our cell sorter in a cassette that allows then the cells to flow in front of 3 lasers. and will thereafter be either positively selected by the rapid opening of a valve that is going to let those cells go in this middle chamber if they do express our marker of choice, PD-1, or let the flow through of cells that do not express PD-1. So it's a novel type of cell sorter that is, cell sorting is done at low pressure compared to the traditional FACS technology, where this valve is controlled with a microchip and can open and close 20,000 times per second, I believe, if I recall correctly, to enable cell selection. And this cartridge is sterile, it's GMP, good manufacturing practice. Once the cells are selected, you can take this cartridge, you bring it under the hood, and pipe it out the selected cells that can then be expanded to large numbers for eventual reinfusion into patient. So we've tested how this new cell sorter could enable us to make a cell product out of CD3 PD-1 positive T cells. You can see here on the left in melanoma tumor cell suspension or GI cancer metastases, the baseline expression of PD-1 out of CD3 T cells, which is pretty similar. Post-sort, we were able to significantly enrich CD3 PD-1 positive T cells with good purity and excellent viability. In this data set, the lowest number of cells we could sort was 3,000-ish, and the highest number was 2.4 million, for a mean of about 500,000 of cells selected from those various solid tumors. Once the cells are selected, we use small bioreactors that are called GREX. They're equipped with a membrane that favor gas, that allows gas exchange and a culture of cells, of T cells at very high concentration compared to traditional flasks. The cells are expanded in media with human serum, anti-CD3 to stimulate the TCRs, IL-2 as a mitogen, and irradiated PBMC feeder cells. We do get very high fold expansion, a mean fold of 12.4 thousand, and are successful at expanding billions of cells for most patients. It's good to be able to use a clinical grade cell sorter to select cells and to be able to expand these cells, but do they recognize autologous cancer and neoantigen? As I mentioned, from the tumor, we do expand those population, the PD-1 positive, the flow through, the PD-1 neg, and we also do a bulk expansion of unsorted cells as a control. We always try to use the same tumor to generate target cancer cell lines in parallel to generating TILs, so that we're then able to do recognition assay of TIL against the autologous cancer cell lines. And we use as a readout of reactivity, de novo upregulation of 4-1BB on CD8 T cells, of OX40 on CD4 T cells, and interferon gamma secretion. That's an example of a melanoma patient where the PD-1 selected cells are represented here, CD4 on the left, CD8 gated on the right, in co-culture without any target. You see no significant expression of 4-1BB or OX40 in those cells. And against the autologous cancer cell lines we've generated from the same tumor we expanded those PD-1 selected TILs, you then see much higher reactivity. Here, 22% of the CD8 T cells do appear to recognize the autologous target. PD-1 negative flow-through cells do not recognize, or almost, there's almost no recognition of the autologous target. And the bulk T cells expanded without any sort of selection leads to a cell product that is not really able, either in the CD4 or the remaining CD8, to recognize the autologous cancer cell line. So that's an example, but we have accumulating data, and this is an incomplete data set, that enriching in using PD-1 as a marker is really reliable in obtaining a cell product that will bear that will be reactive to autologous tumor born both in the CD4 and in the CD8 populations. When we're not able to generate cancer cell lines, we sequence tumors, we predict which mutated gene can generate mutated peptide that binds with high affinity to the patient-specific HLA molecules. We ask a company to synthesize this list of neoantigen peptides. We generate autologous antigen-presenting cells from peripheral blood monocytes, either dendritic cells or B cells. We pulse those neoantigen peptides on autologous APCs and then do recognition assay of TIL and use the same readouts for reactivity. That's an example of a uveal melanoma liver metastasis, where the PD-1 selected cells were able to recognize 4 neoantigen peptides, CD8 cells here in blue, when they're just in presence of autologous APC, do not upregulate 4-1BB significantly and do not secrete interferon gamma. But when they're in presence of the mutated RUVBL mutated peptide pulsed on the APCs, you see that 20% of those CD8 do recognize the peptide and secretes a very decent amount of interferon gamma. That's for the CD8s, and you see here that CD4s can recognize those 3 neoantigen peptides. Not all of them can secrete significant amount of interferon gamma. This is normal in T cell biology because all those T cells are not necessarily Th1 polarized. We only see these types of reactivities against neoantigen when we do select for PD-1 T cells. There's no reactivity in the PD-1 negative flow through, and bulk expansion very most of the time do not lead to significant recognition of neoantigens. So I've shown you data to support that there are ways, there are antigen agnostic ways to select TILs that are able to recognize autologous tumor cell lines and neoantigens. We started this endeavors using a new clinical grade cell sorter, and started this effort by targeting PD-1 high TIL expressors. We also are pursuing another strategy, which is engineering peripheral blood T cells with defined TCRs that are able to recognize neoantigens that are shared across multiple cancer types and ideally presented by a common HLA. We talked about TIL therapy up to now as one of the promising type of adoptive T-cell transfer immunotherapy for solid cancer. But many groups are deploying a lot of efforts in the field of gene engineering to use defined TCRs or chimeric antigen receptors to redirect the reactivity of peripheral blood T cells towards solid cancer antigens. The shared oncogene we've decided to target is KRAS. As you know, KRAS is a common oncogene mutated in a majority of pancreatic cancer patients, tumor in about half of colorectal cancer patients, and 10 to 15% of patients with lung cancer. The most common mutations are on codon 12, the G12D and G12V mutations, and so we decided to focus on these mutations, and we've decided to focus on an eventual peptide that is presented by HLA-A2, which is one of the most common HLA expressed by Caucasians, but not only Caucasians. It's about 40% of Caucasians and 20-25% of Asian patients, for instance, and slightly more in other people. KRAS oncogene is mutated, eventually transcribed as RNA and translated into a mutated protein. And this protein is degraded in the proteasome, chopped into small peptides. And the peptide that contains the G12D or V mutation can be presented by HLA-A2 with a very decent predicted binding affinity. So in collaboration with Rob Holt, we've screened the blood and TILs of countless number of patients in search of TCRs that could recognize KRAS G12D or V presented by HLA-A2. And at the end, we were able to clone out several candidates. The TCR that we want to bring into the clinic is we've called it KTCR1. We're pursuing the preclinical work to make sure that it has a high affinity, a good specificity, and working towards a preclinical trial application with that TCR and cell product. I'm showing you a snapshot of some results we've obtained with this TCR. We can gene engineer human CD8 T cells to express this KRAS G12V TCR and adoptively transfer these human KRAS TCR engineered T cells into immunodeficient mice bearing implanted human tumors. In this case, a tumor that do express KRAS G12V. And you can see after injection of those human CD8 T cells engineered to recognize KRAS G12V that there is some control, slowdown of tumor growth in this model. The specificity appears very good in vitro, and here in vivo, when we implant a tumor that expressed the wrong hotspot mutation, the G12D, you don't see the tumor control as with the KRAS G12V tumor. So thus far, this TCR looks good. We're very happy that HLA-A2, a common HLA, can be targeted. And recently, you may have seen that the proof of concept that KRAS can be targeted with gene engineering has been done by Eric Tran, who works at Providence, Oregon Cancer Institute. Eric had found some KRAS reactive TCRs in TILs in pancreatic cancer patients treated in our GI TIL protocol at the NCI. And this TCR could recognize G12D presented in the context of HLA-C 0802, which is an HLA that is not so frequent in the population. But nonetheless, he showed in this very nice case report published in last June in New England that a pancreatic cancer patient could have control and respond to this TCR after adoptive transfer. I want to close this presentation and leave time for questions. Briefly to talk about what we've put in place at the CHUM to do T-cell immunotherapy for solid cancer patients. The CHUM is a high volume cancer center who is doing autologous bone marrow transplant. There's the largest volume of solid organ transplant here. So there is medical expertise to do T-cell adaptive transfer. We've opened a new early phase trial unit led by Dr. Raima Jamal, which allows us to safely perform those complex T-cell therapy trials. We are 6 scientists that are focusing our lab effort on solid cancer immunology. The research Cancer Access is equipped with an immunomonitoring platform, and Anne-Marie Mesmasson has been a leader in Canada in setting up biobanks that we're using to do a lot of the preclinical and the on-trial correlative research work. The blood bank is supporting this effort, managing also with hematologists a large leukapheresis unit, and with the expertise of thawing cells at the bedside for the bone marrow transplant patients. And we've set up a small cell production unit that I'm going to talk about on the next slide. FACT accreditation, which proposed the best standards for cell therapy worldwide, we've started the accreditation of FACT at the CHUM just before while COVID was on its way. So things are going forward, not as fast as we would like, but going forward. And we're doing all this with key partners. The small cell production unit I'm leading is within the operating room theater, so very convenient to grow TIL for therapy. It's really next door from operating rooms. It runs as a GMP phase 1 compliant unit and should lead to our first investigator-initiated TIL trial using PD-1 selected cells, which I hope we can activate in the early 2023. In our pipeline, we have 4 effector T-cell adoptive cell therapy trials, one that is now closed for accrual. And I hope that the KRAS TCR trial can be initiated at some point in 2023, 2024, in the near future. So in conclusions, adoptive T-cell immunotherapy for solid cancers is undergoing rapid development worldwide. TIL immunotherapy is soon expected to be FDA approved for melanoma, lung, and cervical cancer. Even if neoantigen reactive T-cells are enriched in tumors compared to the peripheral blood, their numbers remain low. And so we need ways to enrich these cells to make better cell product. We're working to develop TIL product enriched in tumor-reactive T-cells by an antigen-agnostic approach based on PD-1 high expression and the use of a clinical-grade cell sorter. We are also targeting the KRAS oncogene with a TCR engineering approach. And I would like to conclude by saying that currently, Canadian patients with solid cancer largely have to go to the US to have access to TIL therapy. And I really hope and really think that we can do better from now on and develop this field in collaboration as much as we can for the benefit of patients. All this work is done by many people. I've highlighted the persons here who have generated the data I've presented. I will be very happy to answer your questions, and I thank you for your attention. Thanks.
Thank you so much, Simon, for this very comprehensive and very interesting presentation. Perhaps I can open the floor for questions. I have Dr. Bass